Today the topic that we are going to start is related to the nucleic acid and protein synthesis, right? In this chapter, we are going to first see about the importance of nucleic acid. That what is the importance of DNA as a genetic material or as a hereditary material. So scientists, when they were making research initially, what they were looking for any of the molecule which should have the ability to store a lot of information in it. Like DNA is the molecule which has a lot of information in the form of genes, and on this, the structure of gene, DNA is instructing RNA to make certain proteins which are actually making certain characteristic of the enzyme, right? And they control the behavior of cell. So different cells make different type of gene, they different type of gene make different type of protein. In this way, their behavior is changed from each other. Like neuron is different from nephron, nephron is different from skin, and a skin cell is different from the muscle cell, right? So number one thing, they wanted to see that a molecule which can store a lot of information in it, that was obviously DNA. And they were also looking for any molecule which should copy itself accurately. And we will see in this chapter that DNA is a molecule of the genetic or hereditary material because it can copy itself accurately. There is a complementary base pairing in DNA. Means adenine is always pairing up with thymine, and thymine and cytosine is going to pair up with the guanine. So in 1940s and other research, they found that this type of molecule can be DNA, right? Because previously they were thinking protein are so important molecule in the body of any cell that they make the dry mass 50% dry mass of any cell, and they are so important that it should make the hereditary material of the organisms. But later on, when they were making research, they found that protein can easily denature itself at a higher temperature. And you know about it कि protein ऐसा molecule है जो कि easily denature हो जाता है high temperature पे. So they discarded this idea and they thought that no, protein cannot be the genetic material for the human or any other organism, because it should be very accurate molecule and it should copy itself very accurately, because the next generation, when they will have the transfer of hereditary material, the cat will make the kittens and the human will make human babies, and dogs will produce their own baby dogs. In this way, they are going to continue their generations after generation, right? And there should not be any mistake, because आपको पता चलेगा इसी chapter में we'll discuss about the mutations or also कि sometime by the mistake of only one base pair there is a very drastic change in the body, और पूरे जीन के structure change हो जाते हैं, and they also cause a lot of change internally, and externally this change may also be visible. So when we talk about the structure of DNA or RNA, generally we call them nucleic acids, and DNA and RNA are the structures which are, wait a minute, I'm switching the screen. So when they were thinking about the molecule of DNA, screen is showing some problem. So again, we will see how the DNA RNA are made up of. DNA and RNA are made up of polynucleotides. Polynucleotide means having, poly means many. Poly means many. So they are made up of many nucleotides. And what nucleotide is? Nucleotide is the monomer of nucleic acid. Monomer or micromolecule of nucleic acids. Means DNA and RNA are made up of nucleotide. So when we talk about the Assalamu alaikum. Actually, I'm busy in class right now. When I get free, I will call you. So, what is the meaning of nucleotide? When we talk about the nucleotide, nucleotide is the general formula of nucleotide. I am talking about general. I am not specifying that it's either DNA or RNA. So keep this thing in your mind when examiner is asking you about the nucleotide. What nucleotide is? You will mention that nucleotide is made up of only three components. And what three components are? Number one is phosphate group or phosphoric acid. Phosphate group. Number two is the pentose sugar. Means 5 carbon sugar. And number three is the nitrogen containing base. Nitrogen containing base, right? This is the general formula for any nucleotide. But when I specify that this is the nucleotide of DNA, then what will I write? See, when I mention that this is the nucleotide of DNA, then here, first of all, I will write one phosphate group, which is the part of both DNA and RNA. Number one. Number two, I will mention deoxyribose sugar. Deoxy means having one oxygen less, deoxyribose sugar. Obviously, it is 5 carbon sugar. And number three, it is any one nitrogen base. Any one nitrogen base. Base out of 4. A, T, C, G. Is that clear? This is the nucleotide of DNA. Now, I am going to erase this general information about the nucleotide. I will specify this side for RNA. So, when I write here the molecule of nucleotide of RNA, then I will not erase this phosphate. It will remain phosphate as it was the phosphate in DNA as well. Then I will write here not pentose sugar. I will erase pentose sugar. I will write here because pentose is the general information, means having 5 carbon. I will erase this information and I will write, although it is a 5 carbon sugar, but the name should be specific because I am talking about RNA right now. I am not generally talking about any of the molecules. So I will separate it and I will write here ribose sugar. It is not having one oxygen less. Full formula of ribose sugar will be mentioned here. Then we come towards the nitrogen containing base. Any one of the nitrogen base. I will erase this. Obviously, it is nitrogen containing base, but the thymine will be replaced by uracil here. Nitrogen base, anyone. Right? Any one of out of the four. A, U. It means A is connected with U and A is complementary to U. And C, G. any one out of these four should be attached with the nucleotide. Then they will be making a complete nucleotide, right? I will repeat. When I talk about the specific nucleotide of DNA or RNA, the first difference is ribose and deoxyribose, number one. And the second difference in their nucleotide is that here in RNA, uracil, and in DNA, thymine is present in place of uracil. So in DNA, A is complementary and making pair with T. In RNA, A is making pair with U. I will erase this information. Note it down, please. Giving you 30 seconds. Here we will see ATP, energy currency of a cell. This is known as energy currency of cell, and it is also known as mononucleotide. Mononucleotide means it is one nucleotide because it is fulfilling all the requirements which should be completed by the definition of any nucleotide. That is why we call it mononucleotide. And mononucleotide means one nucleotide which is having one phosphate group. You can see it is having more than one phosphate group. It has three phosphate groups, not one only. It has a ribose sugar, which is a pentose sugar. It also has one nitrogen base, which is adenine. So adenine, when combines with the ribose sugar, it is known as adenosine. This is known as energy currency because it contains three phosphate group and we call it adenosine. Adenosine means ribose plus adenine. Triphosphate. Tri means three. It contains three phosphate. It is a nucleotide, but it has two more phosphate group. So it is rich in currency. When it loses its one phosphate and provides energy to any of the reaction, we call it ADP. So ATP, adenosine triphosphate, is converted to ADP, diphosphate. Then one more phosphate is lost, we call it AMP, adenosine monophosphate. Mono means single phosphate. When it loses one more phosphate, we call it adenosine. And when we eat something and body is making energy, again they get their phosphate, then adenosine monophosphate, then again one more phosphate, then adenosine diphosphate. Then they get one more phosphate and they make the adenosine triphosphate. In this way, the structure of ATP is formed. So this is a molecule which is very small. It can easily cross the membranes or the different areas. It is carrying a lot of energy. Number three, it is easily convertible, interconvertible molecule. Interconvertible molecule. It can easily switch from ATP to ADP or ADP to ATP. That is why it is easily providing energy or making energy inside the cell. And different reactions which require energy are using this molecule as an energy currency. Then we talk about the two nucleotide. Dinucleotide means when two nucleotides combine together, we call them dinucleotide, like we were calling about dipeptide, disaccharide. So this is di means two. They contain two nucleotides joined together. And the bond which is made in between them, it is not a peptide bond. It is a phosphodiester bond. In glucose, we have glycosidic bond. In glucose, we have glycosidic bond in glucose. In the amino acid or proteins, we are making the peptide bond that is connected with peptide bond. In the lipids, alcohol, ethanol, and the fatty acid, they are connected with the help of ester linkage or ester bond. Here in the nucleic acid, we are having the phosphodiester bond. Phospho in between and both sides connected with the ester bond. That's why we call diamine two ester, two ester bond on both sides of phosphate. So they contain phosphodiester bond, which make the structure of DNA or RNA. The sugar and phosphate are connected to the phosphodiester bond and they make the backbone of the polynucleotide. You can see. Structure of DNA. Watson and Crick were the scientists which were making research, who were making research on the structure of DNA. And later on, they found that there are some complementary base pairing in the DNA. And DNA is having like this structure, which is making a backbone with the help of sugar and phosphate. And when we open the coiled structure of DNA, they have a double helical structure. In this double helical structure, the phosphate and sugar are making the backbone. So when we uncoil, DNA is a helical structure like this. When we uncoil this structure or untwist this structure, they look like a ladder where backbone is made up of sugar and phosphate on both sides. This backbone and this backbone is made up of sugar and phosphate. Number two, DNA is an anti-parallel structure. Anti-parallel means if one phosphate is heading upward, other phosphate is heading downward. Then, one more thing. They contain equal amount of adenine and thymine and cytosine and guanine. They are equal. Why? Because they are complementary to each other. They are standing opposite to each other. Now I am going to tell you in detail that what is the structure of DNA and what is the difference in between the DNA structure and RNA structure. RNA is a single strand. DNA is a double helix. It is also known as duplex. And there is a right-handed duplex. They turn towards the right side, both strands. You can see here, adenine is almost equal to the thymine and cytosine is equal to the guanine. They are showing you the different example of the DNA extracted from the different animals and different plants. Watson and Crick, when they were making research on them, what they have found about their structures, number one, their nitrogen bases are connected with the hydrogen bonding, which make the ranks of the DNA this ladder-like structure like this. If they are making ranks of this ladder-like structure, nitrogen bases. Sugar and phosphate are making the backbone on both sides, and they are anti-parallel chain. Two chains anti-parallel running to each other. Often one turn is containing 10 base pairs. One turn is containing 10 base pairs. So these are the basic information about the DNA. Now we go further and see how it is made. DNA molecule has the following feature made up of two polynucleotide chain. Keep this thing in your mind. There are two polynucleotide chain. Each chain is a right-handed helix, and the two chains are coiled around each other to form a double helix known as duplex. The chain run in an opposite direction, said to be antiparallel. Each chain has a sugar phosphate backbone projecting at right angle, and the nitrogen bases are projecting at right angle to each other. The bases in one chain are attracted to the bases of other chain by hydrogen bonding in between them, and because of the way they fit together, like a jigsaw pieces, adenine and thymine are equal, cytosine and guanine are equal. Now see, this is the structure which I wanted to show you. Look at them. Some of the nitrogen bases are smaller in size, like thymine, cytosine, and uracil. They are smaller. Why they are considered smaller? Because they contain only one ring structure, single ring structure. One or single ring structure. And are also known as pyrimidine. Pyrimidine. Second is the nitrogen bases which are heavier and they contain double ring. These nitrogen bases are only two and they are A and G. They are known as purine or purine. They are known as purine. They are having the double ring structure. They are larger nitrogen bases. So what scientists have made a research about DNA, they thought that the helix of the DNA, when they have uncoiled this double helix structure, which was turning, what they have found that from top to the bottom, like a ladder, they look straight and there is the same width of this molecule. So if we connect a larger base pair with the other one, a smaller one, then they can fit like this. But if we take two larger ones, on one side it will become broader and on other side it will become narrower, like this. But actually in the reality it is a smooth ladder-like structure having the same width throughout. So they realized that some of the nitrogen bases are complementary. So which nitrogen bases are complementary? When they were connecting single with single, like T with C, it was becoming narrow. When they were connecting double ring A with G, they will also become wider. But they wanted to keep it similar in the width throughout, top to bottom. That's why they thought that there might be some complementary base pairing in between the two nitrogen bases. So maybe one is purine and other is pyrimidine. So when they observe the structure, they realize that one purine is connected with one pyrimidine. Means in the whole width, there should be two nitrogen bases only. Two nitrogen bases in the whole width. But rings should be three. But there should be three rings in the whole width. Three rings of nitrogen bases. So they must keep one double ring structure and one single ring structure. That's why they have chosen A with T. This is A with T and C with G. G with C. But someone asked them that what if they switch T with C? Both are single ring structures? They said no. Because C is complementary to G and they have triple hydrogen bond. How many? Triple hydrogen bonds in between C and G. And you can see here, there are double hydrogen bonds in between T and A. That's why they cannot switch T with C. because T is having double bonding, hydrogen bonding in its molecular structure, and if we replace it with C, then C's one bond of the C will be left empty. There will be no one to accompany it. That's why they thought that there must be some molecular structure. And later on, it was clear, this concept was clear that A and T are complementary. Why? Because they have double hydrogen bond, and C and G are complementary because they have triple hydrogen bond. So you cannot switch them this way. So C and G triple hydrogen bond, A and T having double hydrogen bond. But they have also seen one more thing. They have observed one thing. And what they have observed one thing, they observed this, that DNA, when making the structure like this, 1, 2, 3, 4, 5. 1, 2, 3, 4, and 5. In between them, there was phosphate group, like this. On this side again, I make so one phosphate group is present in between them. So this is known as sugar phosphate backbone. But one thing which I want to mention here, look at them. On this side, 3 is the ending of chain. On this side, 5 is the ending of this chain. This is called 5' end. Primer is a small piece where DNA will start replication. So we have a single dash over 5 or 3 which mentions primer. This is 5' end, and this is 3rd primer end or 3' end. One most important thing to tell here, that DNA has a enzyme, which can replicate only from the 3rd primer end. Means replication can take place this way. They don't have any enzyme to replicate it from 5th primer end. That's why whenever DNA will replicate itself, they are going to make it from the 3rd primer end. So if I write it here A, then it is G. It is C and it is T. And they contain the hydrogen bondings in between them, like this. A and T double hydrogen bond, C and G triple hydrogen bond. And which side will be replicating the DNA? 3rd primer end, not 5th primer end, because they are anti-parallel. If one is heading upward, other is heading downward, you can see. This is known as 2 chains of polynucleotide. Why 2 chains of polynucleotide? Look what nucleotide means here. 1 nitrogen base, 1 phosphate group, 1 pentose sugar. It's a nucleotide. It's a nucleotide. So in this chain, they have many nucleotides. On other side, they have many nucleotides. That's why we call that DNA is made up of 2 polynucleotides, 2 chains of polynucleotide. Going further, You can see here. A is always linked by T with T. C is always linked with G. A and G are purine. G and C are pyrimidine. Purine is always pairing with pyrimidine. Purine are two ring structures. Pyrimidine are single ring structure. Sugar phosphate is making a backbone. A complete return is having the 10 base pairs. Structure of the DNA replication. We are talking about the DNA replication. That's what DNA replication is. DNA replication means DNA is going to replicate itself, and when DNA is going to replicate itself, it is necessary that first it unwinds itself like this. DNA first unwinds itself. So there is a molecule which is unwinding it, and this molecule is known as helicase. And the DNA replication is taking place in the S phase of cell cycle. S phase of cell cycle. DNA replication in S phase of cell cycle. So helicase enzyme opens or unzips DNA. It unzips DNA at the time of DNA replication. It opens just like a zip. Helicase open the chains of the DNA. On one side, there is a third primer end. On the other side, there is a fifth primer end. And I already mentioned that DNA is going to replicate from the third primer end. So first of all, a primer will be connected. Primer is a small piece of RNA molecules on which the DNA polymerase is going to connect and replicate, start replication. There are many free nucleotides in the nucleus which make this chain, and DNA polymerase will be connecting itself here. It is DNA polymerase enzyme which make the polymer of DNA. DNA polymerase will make a green line and they will continue to make it. They move in the direction like this. And the part which is made here like this part is known as the DNA fork. They make a fork-like structure. So DNA polymerase start to replicate inward and this chain will going further like this in this direction inward. And this is third primer end. The chain which is made, its replication is continuous replication. So we call it, this strand is continuous synthesis. It is showing continuous synthesis. And it's a leading strand going from third to fifth primer end continuously. We call it leading strand. So what I told you, first of all, DNA is replicated in the S phase. I use another color. DNA is replicated in S phase. What is the enzyme opens it? It is known as helicase, which unzips the DNA helix. Number 3, DNA starts its continuous replication on the third primer end. First, a small piece of RNA, which is pink structure, you can see it's a primer. First, a primer will be connected to the template strand. Template is the DNA. Template means model. DNA is behaving like a model and they are exposing their nitrogen bases according to the nitrogen bases. Like if there is a, they will connect T. If there is a C, they will connect G in front of them. So complementary base pairing is taking place. Free nucleotide are present in the nucleus and they are going to come here. DNA polymerase is connecting these nucleotides and making another chain opposite to the previous one, opposite to the template strand. They are making a new strand, which is mentioned with the green color. And the previous strand was of shocking pink color. So this replication is taking place like this. I have also mentioned that a structure, when they unzip the DNA, structure of fork like structure is made, we call it DNA fork or replication fork. We also call it replication fork. So hydrogen bonding is broken by helicase. Primer is initiating the replication. DNA replicates by the help of DNA polymerase. It makes a continuous synthesis. That's why we call it continuous synthesis, and it's a leading strand. This strand, which is made from third primer side, is called leading strand. Now we talk about the another strand which is on the fifth primer end. This is fifth primer end. On this side, if fifth primer end is here down, it means its third will be upward. So they start to initiate from the third primer end and from the DNA fork. So a small primer will be made here like this. This is the primer. I will use another color. A primer will be made. Primer will be connected because primer is the base on which DNA polymerase will start its working. It's a small piece of RNA base pair. So primer will be connected in front of the third primer end. Then they keep DNA polymerase here. DNA polymerase will be connected here like this. And this is again DNA polymerase. DNA polymerase will make a strand. A strand will be made down on the downward direction, but in pieces, not continuously. This smaller piece of DNA is given a new name on the name of a Japanese scientist, Okazaki. So we call it Okazaki fragments. And Okazaki fragments will not stop there. They will come and join the strand in the beginning. In the beginning, they will stand like this. And again, the third primer end will make a primer, DNA polymerase will make another piece, and the second piece will be connected like this. But still they need to be connected and they need a glue-like enzyme. So this glue-like enzyme will be the ligase. Ligase will be behaving like a glue in between them, and they are joined together. As this in synthesis is not continuous synthesis, we call it discontinuous synthesis because it is